Hello, everyone, and thank you for attending the BIOKDD workshop. I would also like to thank the organizing committee for their hard work in making this event possible. My name is Wenbin, and I am an upcoming second-year PhD student at Carnegie Mellon University. Today, I will be presenting on distance-preserving generative modeling of spatial transcriptomics, a project conducted in collaboration with my colleague, Jing Hong. Transcriptomics analysis is essential for understanding the complete set of RNA transcripts produced by the genome. It provides insights into gene expression patterns, cellular functions, and biological processes. Traditionally, transcriptomics dataset have been obtained through single-cell RNA sequencing. However, as the figure shows, a drawback of this traditional process is that it involves a step where tissue must be dissociated, resulting in the loss of spatial information. The recent advancements in spatial transcriptomics technology now allows us to preserve this spatial information. It not only captures gene expressions for each cell, but also produces 2D spatial information matrices that align with gene expression data. This enables comprehensive analysis of spatially resolved gene expressions. When integrating machine learning models into the study of spatial transcriptomics, it is crucial to develop algorithms that efficiently leverage this spatial information. Recent efforts have studied how to model spatial transcriptomics with machine learning. For example, some studies use graph neural networks, GNNs, where spatial locations are represented as graph nodes. However, these models are often designed for specific tasks, such as spatial clustering, which is shown in the figure here, and do not easily extend to other types of analysis. In contrast, generative models, particularly those based on representation learning, offer great flexibility. They can be adapted for various tasks, including cell type identification, batch correction, visualization, clustering, and differential expression analysis. A key aspect of these models is their ability to extract low-dimensional latent representations from high-dimensional gene expression data. However, unlike GNNs, generative models lack a straightforward way to incorporate spatial information into the learning process of latent representations. This raises an important question. How can we effectively model spatial information when building generative models for spatial transcriptomics analysis? We will explore this challenge later. Within the machine learning community, there has been growing interest in geometry preserving generative models. These models aim to preserve the geometric properties of the dataset. Various approaches, such as isometry metrics and constraint optimization, have been explored. In applications, these models have shown a success in tasks where geometric properties are crucial, such as in computer vision. However, as it is the case with spatial transcriptomics, the challenge of incorporating additional spatial information into these models has not been fully addressed. So we asked the question, how can we integrate geometry preserving concepts into the study of spatially resolved gene expressions? To answer all these questions, I will introduce how we define a distance-preserving generative model. Based on this definition, we'll derive a tractable loss function to enforce our objective, and then validate our approach using the mouse brain tissue's Visium datasets. But first, I'll provide some background on variational autoencoders, which are essential to our study. A variational autoencoder, or a VAE, consists of two parts, a probabilistic encoder and a probabilistic decoder. The encoder maps input data into a latent low-dimensional space, and the decoder reconstructs the data from the latent space. The VAE's goal is to maximize the likelihood of the reconstruction data, requiring the latent representation to capture sufficient information from the gene expression data. This objective is expressed as the elbow loss, and the empirical loss function is written as shown. Here we present preliminary results from applying a vanilla VAE to a sample mouse brain tissue dataset. The original spatial features are shown on the left, while the extracted latent representation is on the right. Note that the vanilla VAE does not utilize the spatial information available in the dataset. So formally, we have two datasets, one for gene expression and one for spatial information. The gene expression data is input to the probabilistic encoder, which outputs the latent representations. However, the spatial information remains unused. However, there's compelling evidence that spatial information should be integrated into our model. As shown in the figure on the bottom right, the colored region represents spatial domains derived from clusters in the reduced dimension space using UMAP on gene expression data. These regions exhibit strong local clustering patterns in the original space. In other words, within each spatial domain, gene expressions exhibit similar properties that are captured in the latent space. This suggests that gene expression is partially represented by spatial information in a neighborhood. This local geometry preserving property is commonly observed in spatially resolved gene expression problems. The challenge, then, is to bridge the gap between the geometric similarities in the original space, depicted by spatial information, and the latent representation space. Now let's move to the main contribution of our work. We begin by providing a rigorous mathematical definition of distance-preserving generative models. Using our earlier example as a motivation, our goal is to ensure that the probabilistic encoder function exhibits a smoothness property concerning both the spatial information space and the output space. For smoothness, one can think of ellipsis function. However, defining smoothness for a probabilistic encoder function is not straightforward. The definition of ellipsis function applies only to deterministic functions, creating a gap when considering probabilistic functions like those used in VAEs. Here, we define a distance-preserving generative model, with ds and dz representing the spatial and latent distance metrics, respectively. S and z denotes the spatial information variable and latent variable. The model captures the geometry-preserving concepts because of the two highlighted parameters. So briefly, with a small L and epsilon, the encoder can generate concentrated latent space samples with high probability, where here L is the distortion concept that measures how unsmooth the probabilistic encoder function is, and epsilon is the error parameter that measures the maximum allowance of the ratio of outliers. This illustrative figure here helps visualize the objective model definition. For four sample cells represented by two blue and two green dots, the closer their pairwise locations, the closer their pairwise distance will be in the latent space. Although we define the model mathematically, this definition doesn't tell us how to train it using data-driven methods. The definition isn't directly optimizable by standard solvers like gradient descent. Thus, a new challenge arises: how to derive a tractable learning algorithm that enforces this definition. In the next section, I will explain how we derive a tractable loss function for our objective and specify the final model. We summarize our results in the following main theorem. Here, C is a constant irrelevant to distortion loss, so minimizing the distortion loss directly minimizes the second term in this bound, keeping L small while the error parameter remains constant. The key takeaway is that minimizing the distortion loss makes the encoder generate more concentrated samples while maintaining the probability of generating these concentrated samples. Another advantage of our theorem is that the distortion loss proposed here is fully decomposable, allowing for easy approximation via multicolor estimation. In our final implementation, we incorporate this loss into the original VAE model as a penalty rather than a constraint. We introduce a weighted matrix G into the distortion loss function. Since the geometry preserving property in spatial transcriptomics is local, the weighted matrix assigns more weight to nearby cells, reducing interference from distant regions. In our experiments, we use a KN algorithm to determine a 0-1 matrix for G. In summary, our model is tractable and easy to implement as an unconstrained optimization problem with a decomposable loss. It is also flexible, allowing for arbitrary VAE models as the backbone. For instance, we incorporate single-cell variational inference, or called SCVAI, in our experiments. So finally, we validate our proposal on 4 real mouse brain Visium datasets, which I will briefly review due to time constraints. In our first experiment, we compared the geometric similarity of the 2D latent representations learned by the SVVI model and its distance-preserving variant. The ellipses represent equidistance mapped from the original space to the latent space. As shown, the variant preserves the geometric structure better than the vanilla model. Enforcing the distance-preserving property empirically improves spatial correlation between the original space and the latent space, as indicated by metrics like Moran's I and Gary's C in our experiment here. Another criterion for evaluating generative models is the mean square error of reconstructed samples on test datasets. As shown, the distance-preserving variant consistently outperforms the vanilla model across 4 datasets in repeated experiments, demonstrating that encoding spatial information can enhance model performance. However, a limitation of our work is difficulty in determining an optimal regularization coefficient for the distance-preserving property. The subplot on the left shows that while MSE may reach low levels, it also exhibits high variance across different covariance values. This may be due to randomness in the datasets, and further experiments are needed. Thank you very much, and please feel free to ask any questions during our Q&A session or contact us via email. Thank you.